Hello children, so in this video let's study some important questions of the chapter biomolecules, and I'm sure these questions will be there in the board exam and you're going to benefit by this video. The first question is define biomolecules and what are the main types of biomolecules. The question was asked in the year 2015 and 19. So biomolecules are organic molecules. Organic means they must contain essentially carbon atom, and they are essential for number one, growth, number two, development, and number three, functioning of living organisms, that is your plants as well as animals. So that is the function of biomolecules. There are four types of biomolecules which are important ones. There are still many more, but the important ones we are covering up here. The first one is carbohydrates. The example of carbohydrates, like for example we have glucose. Proteins, we can have good examples like casein in the milk, we can have myosin in the muscles, we can have keratin in the hair and the nails, and so on and so forth. Lipids, for example, we have oils and fats which are used as reservoirs of energy. And there are two types of nucleic acids which are DNA and RNA. I'm sure you know the meaning of these two, DNA and RNA. Let's write down their full forms. So DNA stands for deoxyribonucleic acid. So it is a kind of nucleic acid. You can see this word. And the second nucleic acid is ribonucleic acid. So the name is quite simple. Let's go to the second question. Explain the structure of amino acid with a suitable diagram. Question asked in 16 and 18. So let's understand the meaning of the word amino acid first. So amino acid, as the name itself indicates, that they contain two important functional groups, that is NH2 known as amine or amino, and carboxylic, that is your COOH. So any amino acid must essentially have these two functional groups, and they may have some other groups attached, for example a hydrogen atom and an alkyl group, that is R group, which is a side chain. Now the simple Fisher projection formula which you can draw in the exam to show is in this manner. You can draw COOH on any one of the sides, NH2 on another side, and on these two places you can draw any two groups like a carbon group, or you can just simply draw hydrogen atom. Now when we draw this compound, when we talk about this carbon atom, this carbon atom is going to be a chiral carbon atom, and the name of this simple amino acid which is important for our exam is glycine. It is the only amino acid which is optically inactive, and for our knowledge we must remember that there are total 20 naturally occurring amino acids in this world, and out of which the only amino acid which is not optically active is going to be glycine. So the general formula of glycine, you can see this is the formula, and the general formula of any amino acid, if you say, it can be simply written by drawing two lines in this manner, writing NH2, writing COOH, writing a R, and writing a hydrogen atom. So this group, the R group, goes on changing, and R actually represents the side chain which will be differing in each amino acid. Let us have a quick look at the amino acids which are important from the board examination point of view. So these are taken from NCERT textbook. Four of the amino acids which I have chosen here. The first one is glycine, and the amino acids are represented by two different methods. Number one is by using 3-letter symbol, or the second one is by using 1-letter code. For example, when we see glycine, okay, when we see alanine, so how do we represent these two amino acids? So glycine can be represented by using the 3 letters GLY or simply G. Similarly, alanine can be represented by using ALA, or simply A. So these are the two shortcuts. Apart from this, you must have seen what there are two asterisks over here. So these two amino acids, that is valine and leucine, are very essential for our growth. So they are called as essential amino acids because they are not synthesized in our body. They have to be consumed in the diet. When we see these two, glycine and alanine, they are simply non-essential amino acids, of course, because they are synthesized in the human body. So from the board examination point of view, please remember the examples of at least two amino acids which are essential and two which are non-essential. The next question we go to is explain the structure of nucleic acid with reference to DNA. So now instead of DNA, they could have also asked you a question of with reference to RNA. Look at the structure. Now this structure, double helical structure, was given by the biologists Watson and Crick. Now from the chemistry point of view, you will be not asked the names of the scientists, but you will be asked questions related to what is chemically DNA made up of. So children, DNA is the standing for deoxyribonucleic acid, and it is a double-stranded helix made up of nucleotides. So the basic building blocks of DNA as well as RNA is a nucleotide. So I can just simply say that if I just go on joining lots of nucleotide like this by certain connections, then this kind of long chain is known as a polynucleotide, right? So many nucleotides coming together is called as a polynucleotide, or simply it is known as DNA, or it can also be known as RNA depending upon its composition. Now when we see that the nucleotide of, let's assume that this is the nucleotide of DNA and this is another nucleotide of DNA, so they are connected by connections. So these connections are important for your board exam. So these kind of connections are called as phosphodiester linkage, right? So please remember this important term, right? What are the nucleotides? How are the nucleotides connected? The answer is they're connected by phosphodiester linkage. So children, now let's understand in detail about what is a nucleotide. Basically, a nucleotide is a unit which is consisting of three parts. Number one, a phosphate group. Number two, a sugar, that is it can be a deoxyribose sugar in the case of DNA, or it can be a ribose sugar in the case of RNA, right? So both DNA and RNA will contain phosphate group, both will contain sugar, but in case of DNA it will be deoxyribose sugar, and in the case of RNA it will be ribose sugar. And the third point is that both will contain any nitrogen containing base. All of these are the bases which are present. So the first one is adenine, standing it A is standing for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. So we are not going to write full names of these bases in the strands, but just represent them by single letters. All right, so the strands, as we talked about, they are double helix, the two helices are intertwined like this, and they are connected by hydrogen bonds. This is important for 1 mark questions in the board exam. So if I write adenine on the left hand side, of course I'm going to write thymine on the right hand side. Similarly, if I'm writing G on this side, I'll be writing C on that side, C on this side, G on that side, and so on. And these bases are going to be connected by hydrogen bonds. So this is the way we are going to connect. Can you see that? So how many hydrogen bonds are present between adenine and thymine? So there are 2 hydrogen bonds, and between guanine and cytosine there are 3 hydrogen bonds. Same way here, I'll be drawing 3 hydrogen bonds. So in the board exam you can draw such beautiful figures, very simple figures with a pencil, and show that yes, you know how the connections are. So let's come back. So we have what is a nucleotide? So a nucleotide in terms of mathematics, let's say if you want to express it, it's a unit which consists of base plus sugar plus phosphate unit, right? What are the bases? The bases are A, T, G, C in the case of DNA. And if you talk about RNA, the bases in RNA would be A, U, G, C. Alright, so this is important for your board exam. So in the case of RNA, thymine is replaced by uracil, right? So there is no thymine in the case of RNA. Third point is there will be a sugar, there will be a sugar also, as I told you, deoxyribose in DNA and ribose in RNA. Let's understand the major differences once again. This is one of the ways in which you can show how a nucleotide looks like. So this is how I've drawn. You can see that. So here the first part which I'm drawing, showing it by a circle, is the phosphate group. Then I'm showing the second part is a base. This is the blue color one, that is the nitrogenous base connected with the carbon number 1 over here. And this is going to be carbon number 2, this is carbon number 4, and this is carbon of 3, 4, and this is a 5 one. So this is the sugar. The yellow one is a sugar. So which sugars are there? Just let me show you once quickly. So as we can see that it's a 5 carbon containing sugar. So now let me draw first ribose, D ribose. So what's the meaning of D? You have understood. D means the OH on the last chiral carbon atom so is on the right hand side. So of course this is that carbon atom which I'm talking about, this carbon atom. So drawing the structure is extremely simple. It's just like glucose. See, we just have to draw CHO on the top, and at the bottom we have to draw CH2OH. Then you can just go on writing HOH, HOH, and again HOH. Children, see, such a simple structure is this. I'm sure your teachers might have taught you, and you might have understood that time also. So bacho, this is D ribose and this is an RNA. So when we look at the numbering, 1 2 3 4 and 5, so it's a 5 carbon containing sugar. Same way when you draw the deoxyribose, so in the case of deoxyribose, what you will do is the second, it's going to be 2D-deoxyribose in the case of DNA. So why it's going to be 2D-deoxyribose is because at the second carbon atom this oxygen atom will be missing. So when you look at the structure, this OH, when you look about this OH, so when you look at this OH, you will remove the oxygen and you just write the hydrogen over here, and the rest of the structure you just draw in the same way as you wrote in the case of RNA sugar. So this is how the 2D-deoxyribose looks like. So this is the one thing. So in the board exam, simply you can draw this kind of figure which we have shown here, this figure. So you can show this figure and show what is the meaning of a nucleotide. Then we can also show diagrammatically how these people have shown here that adenine is bonding with thymine with 2 hydrogen bonds here, and guanine is bonding with cytosine by 3 hydrogen bonds. So here 2 hydrogen bonds, here 3 hydrogen bonds, and so on. Now sometimes they ask you some interesting questions of this pattern, like they will give you 2 strands of DNA and they will give you a sequence of base on the one strand. So and they'll ask you what is the sequence of base in on the other strand. So that's quite simple, because that there is a base pairing principle according to which the bases are going to combine only in some specific way, that is A bonds with T and G bonds with C. So with this logic you can start drawing the the bases on the other side. Like if you can see what I'm doing is I'm just writing the sequence over here. Now C will come, right? So this is how the sequence can be written. Now so the point which we covered up here is that DNA is a double helix, and it shows the sugar phosphate backbone and complementary bases, and the 2 strands are held together by hydrogen bonds. Sometimes questions are asked on the differences between DNA and RNA. So I think we must remember at least 2 to 3 differences. I'll just jot down few differences. Now DNA is number 1 present in the nucleus, whereas RNA is present in the cytoplasm of the cell. DNA is responsible for the transmission of genetic characteristics from the parents to the offsprings, whereas RNA is responsible for the synthesis of protein, right? This is the point number 2. The point number 3 would be the structure, where we see DNA as a double helical structure, whereas RNA is going to be a single helix. It's got a single helix. Then we also talk about, you can say, the bases which are present in DNA are ATGC, whereas in the case of the RNA, you can say the bases are not ATGC, they are AUGC. You can also talk about the sugar present here. We have the deoxyribose sugar present in the case of DNA, whereas in the case of RNA we have the ribose sugar. Yeah, I think that's quite sufficient for you to explain your answer in the board exam. Let's come to the next question. Discuss the structure and functions of carbohydrates. So children, as we already have understood this, we have already discussed this. What are carbohydrates? They are they are optically active polyhydroxyaldehydes or ketones, right? Or the compounds which upon hydrolysis yield such compounds. So the point is, there are two important things over here. They are optically active. Polyhydroxyaldehydes or ketones, or the compounds which upon hydrolysis give such compounds. So now let's see the simple definition over here. Carbohydrates are made up of three elements, primarily carbon, hydrogen, and oxygen, and they can be simple carbohydrates. They are the simplest one, which cannot be broken down further, and the complex carbohydrates, which can be broken down further. The functions are monosaccharides like glucose. They are an immediate source of energy for us. The moment we eat glucose, it mixes with the blood and gives us instant energy. The example is glucon D, commercially sold. It's a glucose. Polysaccharides like starch, which is like for example in potato, you're making bread, rice, etc. And glycogen, which is present in the liver and the muscles, it's a storage, it stores the energy, reservoir of energy. Cellulose, it's a very important structural material for plants. It is used for making the the stem of the tree, or the trunk of the tree. So we look at this. So this will be basically cellulose, this part. So now let's look at the diagram of D-glucose, which we've already discussed. Glucose is a straight chain molecule. It has 6 carbon atoms. It has optically active carbon atoms, as I can show you, 1, 2, 3, 4, and out of which the second last carbon atom, that is this carbon atom, is going to decide the D and L. And as you can see, this is on the right hand side, the OH is on the right hand side, so it's a D-glucose. This kind of formulae, Fisher projection formula, means in the form of plus. You're drawing the vertical lines and you're drawing the horizontal lines. So these kind of formulae are called Fisher projection formulae. So children, how does glucose exist, and how does it cyclize, and all such more points we will see in the next video. Till then, keep studying. There are exams that are approaching, and you must dedicate your time towards proper revision. There are hardly much chapters in your syllabus. So systematically, if you start doing, I'm sure you will score good marks. In case you feel that you require some particular concepts to be explained by me, do let me know in the comment section. Thank you so much. God bless.